Good afternoon. I'm Jae-young Yang, the CEO of Prestige Biologics, and pleased to introduce the company to the virtual audience. To simply summarize who we are, the Prestige Biologics is a contract development manufacturing organization of biopharmaceuticals. We can provide customized solutions to a broad range of customers, from small biotech companies to big pharmaceutical companies. In the time we have together, I will go more into detail about who we are, our unique capabilities, and co-development project and current project. To start, let me take you all through some major milestones of Prestige Biologics since its establishment in 2015. Since 2015, we've been partnering with our affiliate company, Prestige Biopharma in Singapore, to co-develop Herceptin and Avastin biosimilars. In 2018, we opened our first campus that houses our labs and production facility. The campus is located in Osong, which is about 130 kilometers south of the nation's capital, Seoul, and part of Cheongju in Chungcheongbuk-do, and it was warmly welcomed by the city as an addition to the growing biopharma hub here in Osong. The next year, 2019, was a big year for the company. MFDS, the Korean Regulatory Authority, certified the facility as a GMP compliant. We then wasted no time and manufactured our first antibody biosimilar product for the clinical phase 3 trial. Also, in 2019, Prestige Biologics supported Prestige Biopharma in preparing the application for marketing authorization of HD201, a Herceptin biosimilar in Europe. Early this year, the company broke ground to start construction of our Campus 2 facilities. This is our current organization chart of Prestige Biologics. There are 4 main departments: quality, manufacturing, technology and engineering, and administration. The quality department works closely together with the manufacturing to ensure all steps of manufacturing process, from product sourcing to testing and monitoring, are performed within a GMP environment. Within the technology and engineering department, our process development team is responsible for all the developmental works to execute manufacture, while the engineering team is in charge of construction and the physical changes to the facility. The admin department manages all things business operation. What you see here is our current and future campuses. The top half is the Campus 1. Currently, Campus 1 has 3 2,000-liter single-use bioreactors, adding up to a total production capacity of 6,000 liters. What makes Campus 1 stand out from others is that it was the first production facility in Korea to adopt a single-use system for the entire manufacturing process. The bottom half covers the additional facilities currently under construction or in planning. The photo in the left is a Campus 2. It's not too far from the Campus 1 in Osong. When it is completed, it will have a total production capacity of 98,000 liters using a smart hybrid system. As an extension of Campus 2, we'll be proposing a dedicated vaccine center with a capacity of 80,000 liters. The photo on the right is in the plan to append to Campus 1, an annex with a capacity of 24,000 liters. After completion of all construction, Prestige Biologics will have over 200,000 liters in production capacity. Looking at our domestic competitors and their capacity, This would put us as the second largest CMO in Korea. As you can see from quick turnaround in the Campus One timeline, as a company, we are very driven to make progress and aim to make the impossible possible. I spent a little time explaining more about the Alita, the smart hybrid system that will be implemented in Campus 2. Where while a single-use system offers benefits such as minimal cleaning, sterilization, flexible infrastructure, fast turnover rate, and a minimal cross-contamination, the system has a limitation of scale, low-level automation, and the high expense of consumables compared to the stainless steel system. Therefore, our engineering team was tasked with, is it possible to get a best of both systems? If so, then how? The team came together, take first-hand insight, and came up with a great hybrid system for the culture and purification of antibody drug manufacturing. This new concept was defined and developed proprietary technologies. Technologies include sterilization filter system, aseptic connecting system, and buffer transfer system. We are very excited about this. Over time, it is projected that Alita will increase over 50% in production batches and reduce about 14% in consumable expenses. We've achieved cost competitiveness and highly optimized development and manufacturing process with the key technologies you see listed here. Some to highlight that are critical in the drug development phase are the patented bicisterine expression vector for developing antibody expression cell line and the patented antibody purification method. This enables the manufacturing of a high purity and high quality pharmaceuticals that is cost competitive. Prestige Biologics has not only the key technologies for drug development, but also technologies for commercialization. This includes the key components in the Campus 2 hybrid system mentioned in the previous slide. Now, I'd like to spend time going through a crucial aspect of a pharmaceutical manufacturer, quality system accreditation, or GMP compliance. The facility is recognized as a GMP compliant in accordance with domestic and EU standards. We see the preparation for EU GMP compliance as a high achievement and would like to share why we needed this and how we met the standard. In 2018, the first batches produced at the newly built Campus 1 was a clinical batch of HD201. At the time, it was an investigational drug of the ongoing clinical 3 trial in Europe and required an EU QP release. Through a qualified EU QP audit, we got the authorization to release HD201 from Campus 1. We replicate the process in 2020 and also got authorization for second drug product, HD204. The next task was preparing for the EMA GMP inspection supporting HD201's application for product registration. We hired a global quality consulting firm PQE and invested months preparing in forms of gap assessment, remediation, and mock inspections. By the final mock inspection, Prestige Biologics was deemed suitable for EU GMP. In this next section, I will share our strengths and capability as CDMO. We are building a one-stop shop that sells services from process development to commercial production. Our service include custom engineering for process optimization, productivity improvement, and cost reduction. The full scope of capability across the drug manufacturing lifecycle allows us to provide customized services for every customer type and development stage. We are also able to offer a customer dedicated production facility with an optimized process system and adjusted the customer's product. This here is a table overview of our technology and competitiveness and achievement at each stage until now. In addition to the general CDMO services, like research and manufacturing process development, clinical drug production, licensing cooperation, and commercial production, Prestige Biologics can offer engineering services. Starting with our capabilities in research and process development stage, we have a bispecific vectors technology to establish a solid antibody production cell line with a high titer. We can apply this vector to the customer's antibody gene to create a stable cell line. While product and process assessment, culture and purification process development, and analytical method development are also offered by other CDMO companies. Our strengths are in the flexibility from single-use system and cost reduction technologies. Prestige Biologics can to meet the request of customers, developing product with a complex manufacturing process that's not otherwise possible with a stainless steel system. Next, Prestige Biologics engineering capability. Campus One is the achievement of the engineering team and commendable as it was their first project. After thorough considerations, the team carried out the design of facility for proportional CMO, including production site, QC labs, and utilities according to GMP guideline, and also selected production equipment that can result in best outcome for each product through manufacturing process analysis. Let me briefly describe an antibody production process to give you an idea of how our process flow and the types of equipment we have. The general production process consists of a culture, recovery, and purification. The culture process starts with a vial of cell bank. The cell culture volume is increased to up to 2,000 liters through multiple steps of a seed train. Once the main culture is ended, the culture medium containing the expressed antibody is separated from the cell. These antibodies undergo the following purification processes: 2 or 3 column purification, virus removal, and extrafiltration. We use single-use bags during these entire processes. At the end of purification, we get the final drug substance. The drug substance is formulated to make the final drug product. The purpose of quality control is to ensure antibody drugs are uniformly produced every time, and the entire process from incoming raw material to final drug products are GMP compliant. To do so, antibody drug substance undergoes various tests to confirm the product's property, content, identity, biological activity, impurity, and so on. All meet the predefined product specification. The manufacturer services Prestige Biologics offered include support for clinical study readiness. In the drug development cycle, after the discovery stage, the drug proceeds to clinical studies. Specifically, to develop a drug for human indication, clinical study is essential to collect toxicity, safety, and efficacy data. However, many biotech companies cannot produce their pharmaceutical product for a clinical study. This could be due to lack of manpower, experience, established quality system, or physical facility. This is where Prestige Biologics comes in. In this list of documents required in the clinical trial application in Korea, you can generate and provide document number 4, compliance with the GMP, and number 6, quality documents for investigational product. Those documents are associated with the manufacturer, specification, analytical method, and test result, and stability data, etc. Another capability related to the document generation is supporting the product registration package. The Common Technical Document, shortly CTD, is an application dossier for drug registration in EMA, FDA, and MHLW. Within the Common Technical Document, Prestige Biologics has the experience to generate Module 3 quality document for EMA registration of our co-developed product, HTD-201. This extensive section covers in detail the physical and chemical properties of the drug and the quality-related content of main ingredient, the manufacturing method, quality analysis method, validation standard, packaging material, and many others. Moving on to Chapter 3, the co-development products and current project. I will cover each product in our portfolio. This is HTD-201, a step-in biosimilar with indications for breast and stomach cancer. Its estimated market size in 2019 was about $6.1 billion US. As a co-developer of HTD-201, Prestige Biologics completed the development of process scale-up from 1,000 liter to 2,000 liter, clinical production, process validation for commercial production, and process changes. Phase 3 clinical study results showed drug safety and comparability to the original reference drug. Among its biosimilar competitors, it has the closest clinical efficacy to the reference drug. The EU marketing authorization submission is currently under evaluation, and it's projected that commercial manufacture would begin late this year. Also, a co-developed pipeline HTD-204 is an Avastin biosimilar with indications for colon, lung, and ovarian cancer. Its estimated market size in 2019 was about $7.12 billion US. Like HTD-201, Prestige Biologics has developed a manufacturing process for the clinical and commercial production, as well as analytical method. Currently, the global clinical phase 3 trial is ongoing. Moving on to 2 products under our full CDMO services are PBP-1510 and PBP-1502. PBP-1510 is a novel antibody drug developed to target PALP, a protein widely associated with pancreatic cancer progression and metastasis. Last year, PBP-1510 was designated as an orphan drug by FDA, MFDS, and EMA. And this year, an IND was submitted for a clinical phase 1 trial. Next, PBP-1502 is a Humira biosimilar with indications for rheumatoid arthritis and psoriasis. Prestige Biologics will be primarily responsible for the manufacturing process development, manufacture, and document generation for the clinical phase 1 trial. From this presentation, key takeaway I want to highlight about Prestige Biologics are that we first have a manufacturing competency under a global GMP-compliant infrastructure. Second, our technical strengths from patented innovations such as non-protein purification and the manufacturing system and the use of alternative culture media overall improves cost competitiveness. Third, we've achieved production competitiveness with our Alida Smart Biofactory system to maximize the efficiency and flexibility of the facility. And lastly, our business model offers comprehensive services from development to commercialization for both domestic and global biopharmaceutical companies. Prestige Biologics was founded with one mission in mind, and this is to break the barrier of drug access by providing price competitive yet quality product through optimized manufacturing. Every day, we are making incremental progress and constantly growing from our learnings. We strive to push the boundaries of creativity and innovation to be a leader in the global biopharma industry. If you have any inquiries regarding our CDMO services, we'd love to get in touch. Thank you for your time. Thank you.